Hello, ladies and gentlemen. Before I start, please allow me to express my gratitude to the organizers of the Canadian Poultry Research Forum, who allowed me to share some results and future directions of my research project, the effect of barn sanitation on gut microbiota of broiler chickens. I'm Yi Fan from the U of A, and I'm a PhD student co-supervised by Dr. Corford and Dr. Willing. Let's start with some background of the project. The way that we raise chicken today is very different from 70 years ago. We used to house a handful of birds in the backyard and wait for their eggs. However, today, broilers are raised in commercial barns like this with highly controlled temperature, humidity, ventilation, feed, and water supply. In a common commercial barn like this, 12 to 20,000 birds can be housed together. In this sense, we have changed the living environments that the birds have been adopted for millennia. It is true that the living environments of chickens are greatly enhanced now. The poultry industry still has great challenges. Diseases like necrotic enteritis have been haunting the industry for decades. It is even more challenging for the industry to survive from the global tendency of antibiotic-free farming. Although there's been great effort to minimize the usage of antibiotics, however, they are still frequently being used. Therefore, antimicrobial resistant gene is now a major concern to both academia and the general public. One possible solution may be helping the birds to develop a diverse and balanced gut microbiota. Studies on different animal models suggested that commensal microbes can increase host nutrient utilization and feed efficiency. In addition, commensal microbes are also important in pathogen defense by competing with pathogenic bacteria. Moreover, studies also have shown that in early life, symbiotic bacteria exposure is important to program host immune responses and therefore improve gut health. To better understand how chickens chickens react to early bacterial exposure, some studies looked at the recycled litters. They found that reused chicken litter has an impact on the chicken early microbiota development. One research found that recycled litter has positive and persistent effect. It increased the predominance of Faecalibacterium, a butyrate producer in the ceca of both young and mature birds. It was also found that the intestinal immune responses of birds grown using recycled litter is suggestive of immune activation. However, there are limitations of these studies. First, these studies only focus on one experimenting cycle. Second, these experiments were limited to lab scale. And third, very little attention was paid to foodborne pathogens like Salmonella and Campylobacter. But most importantly, in reality, reusing chicken litter is not allowed in Canada. However, water wash and full sanitation are widely used by the chicken producers. Full sanitation requires high level of chemical disinfection, whereas water wash without disinfectants may leave some beneficial microbes from the previous flock that can help the newcomers to establish their gut microbiota. Knowing the limitation of the previous studies and the barn cleaning regulations in Canada, we want to compare full sanitation and water wash method on the production level. The ultimate goal of this study is to validate water wash method as a reliable or even a better practice for the industry with scientific evidence. In the short term, we want to understand how the cleaning methods affect chicken gut microbiology, pathogen load, and intestinal health. To achieve this, we did our study on 7 similarly constructed barns for a total of 4 flocks per barn. Barn cleaning schedule is as listed. As we can see, we have 14 flocks assigned to each treatment. More specifically, for the full sanitation, we try to keep the barn as clean as possible on a production level. After the removal of used litter, a commercial chemical disinfectant was applied, followed by multiple times of water rinse and fresh litter placement. For the water washing barns, used litter was removed. The chicken house was then water washed, followed by fresh litter placement. Chickens from both treatments were raised in the same manner by the chicken producer. Chicks were placed at day 0 and reared until day 30. 5 birds from each barn were randomly selected for sampling. Cecal content and tissue were collected for gut microbial community profiling, pathogen detection and abundance quantification, as well as short-chain fatty acid analysis. Now we can test our hypothesis that the water wash method impacts the chicken gut microbiota that affects pathogen abundance and the cecal short-chain fatty acid profile. In the poultry industry, foodborne pathogens Salmonella and Campylobacter are major concerns. Therefore, cecal digesta for day 30 birds were used to do a pathogen enrichment assay. Here, occurrence score is defined as the pathogen-positive birds divided by the total birds sampled in the same barn. For example, if 4 out of 5 birds in the same barn were shown Campylobacter positive, the Campylobacter occurrence score of that barn would be 0.8. In our study, no birds were shown Salmonella positive in the enrichment assay, which is good. Campylobacter-wise, on day 30, birds grown from the water wash barns have less Campylobacter occurrence. Since enrichment assay cannot quantify pathogen abundance, qPCR assay was used to quantify cecal total bacteria, Campylobacter jejuni, and Clostridium perfringens targeting 16S rRNA gene, Hippo gene, and NAPB gene, respectively. As a result, compared to the full sanitized group, the water wash group has lower abundance of Campylobacter jejuni at day 30, whereas no differences was observed regarding total bacteria, meaning that the decreased Campylobacter jejuni observed in the water wash treatment was not a result of reduced total bacteria. Furthermore, low level of Clostridium perfringens were observed in both treatments. We have also used 16S rRNA gene sequencing technique to profile the chicken gut microbial community. Our results shown that the cleaning methods did not affect the day 30 alpha diversity indicated by KL1 and Shannon indices. It suggested that the barn cleaning methods did not change the overall microbial species richness and evenness in the chicken gut. Now let's have a look at the beta diversity. Beta diversity considers how similar the microbiota are between each individual. The more similar they are, the closer they cluster together. Results show that the birds from the two different treatments tend to harbor different microbial composition. However, because the R-squared value is very low, it indicates that the difference of microbiota between the two cleaning methods is significant but subtle. Microbial composition-wise, changes of the relative abundance of some microbes were observed on the genus level. On day 30, Helicobacter is more abundant in the water wash birds. The role of Helicobacter in poultry still needs to be studied. Yet in human research, Helicobacter is considered a missing microbe of the microbiome, and it's proposed to be an important commensal. Moreover, we have also found that Bacillus has higher abundance in full sanitized birds compared to the water wash ones. Since Bacillus is spore-forming and can survive from harsh chemical environments, it is not surprising for us to see them being more predominant in the full sanitation treatment group. We have also found that Anostipe was more abundant in the full sanitized birds. However, so far, very little is known about this genus. That means what role they play in the microbiota still needs to be further studied. We have also measured the cecal short-chain fatty acid concentration between treatments. As a result, the water wash group showed increased total short-chain fatty acid in their gut. Specifically, acetate, propionate, and butyrate concentration were significantly higher than the full sanitized ones, whereas same trend were shown on volatile concentration. As short-chain fatty acids are beneficial to host immunity and gut health, it indicates that the water wash treatment may have helped the birds to develop more robust immune responses and better nutrient utilization. Together with the microbiome results, it is reasonable for us to assume that the cleaning method had an impact on the chicken microbiota, which led to an altered short-chain fatty acid profile in the chicken gut. To summarize, there are a few take-home messages from our results to date. At day 30, chickens from water wash barns have less abundance of Campylobacter jejuni. Second, barn cleaning methods have a modest but significant impact on the day 30 chicken gut microbiota. Third, our results show that the day 30 birds from the water wash group have higher short-chain fatty acid concentrations in their gut. So in this sense, barn cleaning method have some interesting impact on the abundance of some important microbes in the chicken gut that influence the Campylobacter jejuni abundance and the chicken gut short-chain fatty acid profile. In the future, we want to see if cleaning methods have impact the antimicrobial resistant gene in the chicken gut. Previously, studies on environmental sciences reported that the environmental stress, such as chemical disinfectants to the bacteria, can trigger antimicrobial resistant gene mutation and transfer. However, very limited information is known about chemical disinfectants and their effect on livestock farming. That opens the window of our study. We will use metagenomic sequencing technique to study the chicken C-col microbes. Ideally, it'll help us to reveal how chemical disinfectants affect antimicrobial resistant gene diversity, abundance, and transmission in the chicken gut. I would like to thank Alma, the Alberta chicken producer, and the Canadian Poultry Research Council for the funding support. Also, I would like to thank my supervisor, Dr. Corver and Dr. Willing, our collab partner, Dr. McMullen and Dr. Ingalls, and of course, all my supportive colleagues. Last but never the least, I would like to thank each and every one of you here for your attention today. Thank you very much.
Oh, all right. Well, thank you very much, Edward. So there are a couple questions in the Q&A, so we'll just go there directly. Question number one, could you specify if water temperature was taken into consideration, and if not, why?
That's a very good question. So it was taken into consideration. So we did our animal trial. It's a year-long trial, so it covers both summer and winter. For the water wash, I think the chicken producers set their temperature, water temperature, between 30 to 35 degrees Celsius. That's a way to limit the variation, because we have extreme weathers between summers and winters. So that's one easier way to control the variant. And we can't set it too high because microbes are sensitive to temperatures too. So yes, water temperature was taken into consideration.
All right. And the next question is, do you think that the beneficial effects of water wash depends on the pathogen load before cleaning?
From our study, we saw a decrease of Campylobacter, but it's a tricky question. It's still limited to our study. We do want to see more study on this field, and before then we can't give any suggestion to the production, to the industry. But yes, we did see decrease on the pathogen load.
Right. Yeah, it might be a relative assessment might be more informative. Another question here is, bacterial isolation can vary by methods of enrichments utilized. Did you use different enrichment medias for specific pathogen type?
Yes, we did use specific media for different pathogens. But when it goes to, well, we just want to be more specific when it goes to the strain level. So we use qPCR as well.
Okay, good. And one last question, and then we'll move on to our next presentation. Was there variation in the source of the chicks? Were they from the same hatchery?
They all were from the same hatchery. There's no differences on the strains.
Oh, it's good to know. Okay, I We do have a couple more minutes, so if you're up for it, we can ask you a few more questions. I was interested to know, so you identified that the fatty acid profiles were different, and that could have some effect, but I was waiting to hear how that linked to their production performance. And did you see differences in production performance?
The production performance data was not was not given to us by the producers. That's one thing that we need to further follow with the producers. Whereas, however, we can connect the microbiology of their of the bugs in the gut and the short-chain fatty acid production level in their gut, and we can draw some correlations between them, and that will well, if we have time in the future, I can present that data. And yes, you pointed out quite well that we need that production performance data from the producers.
Perfect.